In this video, we will discuss about this pancreatic cancer treatment approach using the combination therapy. So here the paper starts by understanding the relationship that is already published in the literature about this RAF1, EGFR and STAT3. So from literature they are able to find out that when this RAF1 and EGFR are ablated, it will cause the accelerated phosphorylations of this STAT3, and that will help in the proliferations of this pancreatic cancer cells. So they wanted to check whether these things they are able to see the same relationship when they do these experiments on the pancreatic cancer cells or not. So what they did is they first prepare a system where they can selectively knock down RAF1, EGFR or STAT3. So this for the knockdown of RAF1 and EGFR, they use this CRE-based inhibition, and for STAT3 they used the silencing RNAs against STAT3, and they check whether the system is working or not. So they checked in two different independent systems, that when they induce this CRE, they are able to see knockdown of RAF1 and EGFR, and when they induce this when they use these silencing RNAs against STAT3, they are able to see knockdown against the STAT3, and when they use both of them, they are able to see knockdown of both. And these things they have checked for two different independent systems, and they are able to see the expected results. Thereafter, they now wanted to see whether it has any impact on the cell death or colonies formations or not. So they use for the control of this CRE, they use this GFP based inductions, or they use GFP based signals. So when they use the control shRNA where the STAT3 is not knocked down, they are able to see a high colony formations. In this case, all the three genes are present, that is RAF1, EGFR and STAT3. And in the case where STAT3 is knocked down but RAF1 and EGFR not knocked down, in this case also they are able to see high colony formations. But in the case of this RAF1 and EGFR knocked down but no knockdown of STAT3, colony formations goes down, but still they are able to see the colony formations. But when all the threes are knocked down, they are able to see very minimum colony formations. So now the question arises, what happens in this case that leads to the high colony formation, or that prevents the cell death? So when they check from literature, they are expecting that this knockdown of RAF1 and EGFR will enhance the phosphorylation of the phosphorylation of the STAT3, and that will provide resistance against the pancreatic cancer cells. So they use the HNA staining to validate, to check this relationship between RAF1 and EGFR and phosphorylation of STAT3. So in the case where RAF1 and EGFR are present, they are able to see a less phosphorylated STAT3. But when this RAF1 and EGFR are knocked down, they are able to see accelerated or enhanced phosphorylation of the STAT3, indicating that this enhanced phosphorylations might prevent or might be the cause for providing resistance to the pancreatic cancer cells against the cell death. So in order to validate that hypothesis, what they did is they use this kind of system and induce the isoform of STAT3 and check whether it can the cells are able to regain the resistance. Pancreatic cancer cells are able to regain resistance against the cell death. So first they validate that they are able to induce this STAT3 and the isoform of STAT3 or not. So this is the control where there is no STAT3 and the phosphorylation STAT3 signals are not there. But when they induce the STAT3, they are able to see enhanced. They are able to see the phosphorylation of the STAT3, and they also checked it for two different conditions, and they are able to see in both the cases the phosphorylation STAT3 signals are again re-established. And then they check the colony formations of these conditions. So when they don't have this isoform of STAT3, they are able to see very minimum colony formations. But when they have this when they induce this isoform of STAT3, they are able to see the enhanced colony formations. and one of the reasons could be because of the enhanced phosphorylations of this STAT3, isoform of STAT3. So this further confirmed their hypothesis that the STAT3 is the one that provides resistance to the pancreatic cancer cells against the cell death. But in the ablation of this RAF1 and EGFR. So in this figure they just confirm the relationship that are all published in the literature about RAF1 and EGFR and STAT3. In the next video we will see how they use this relationship to develop these combination therapies. What are the results that are presented in this main figure. So that's all for this video. If you need to understand more about this topic in detail or about these experiments in detail, you can go through the reference that has been provided in the description. Thank you for your time.